Hi, hello, vanakkam. I am Dr. Vijayan, Assistant Professor of Microbiology, Faculty of Medical Health Science, SRMISC, Trichy campus. Today, I discuss about gram staining. Staining is the artificial coloration of substance to facilitate the examination of tissue and microorganisms or other cell under microscopic view. As initial work with the stain process was performed, some of the microorganisms, example Klebsiella and Streptococcus, he did not use to counter stain. Example, the grams not used counter stain is a procedure. It was a few years later, the German pathologist Karl Weigert, 1845 to 1904, from Frankfurt had a final step of staining with safranin. Hans Christian Joachim Gram, Danish biologist and bacteriologist also, and physician also, developed gram staining techniques in 1883 and published the finding in 1884, the Fried Lanter's Journal. He was working with respiratory disease and lung tissue from cadaver at Municipal Hospital, Berlin, and it is accidentally spoilage of glucose iodine over lung tissue section lead to the development of gram staining. Well, they examined the lung tissue from the patient who had died or dead of pneumonia. He observed that certain stains were performedly taken by the retained by the bacterial cell. The gram staining is a quick procedure used to look for presence of bacteria in the tissue sample and to character the bacteria gram positive or gram negative based on the chemical and physiological properties of the cell wall. This is the cell. The gram staining is the strain the bacteria. The gram staining should be almost always be done as the first step of diagnosis of bacterial infection. He is the famous scientist, the Gram. Example, the Hans Christian Gram. The gram staining is named a Danish scientist. The period of lifetime 1853 to 1938. He developed the techniques of 1882 and published 1884. The famous journals. The two bacterial similar, the clinical symptoms identified: Streptococcus pneumonia, so pneumococcal infections. Another one is Klebsiella pneumonia, bacterial cells. And the Karl Weigert already told 1845-1904 is a lifetime. The Gram did not use counter stain. And after so many years, the German pathologist Karl Weigert had a final step of safranin stain. So safranin is a pink color. The gram staining, it is one of the most important staining method in bacteriology. It differentiates bacterial species into two major groups. One is gram positive bacteria, another one is gram negative bacteria. The help of gram staining, the bacteria will differ due to the difference in cell wall composition. This is the cell wall composition is a gram positive wall and gram negative wall. Right? The thickening of the cell wall is very important. This is a differential stain of gram staining. One is crystal violet solutions, grams iodine solution, 95% ethyl alcohol, safranin solutions. These are the basic requirements. The gram positive bacteria have thick wall of peptidoglycan layers. Peptidoglycan is a polymer of sugar amino acids. Between the cell wall and cell membrane is a membrane teichoic acid, is a very thin layer of lipopolysaccharide is present. This wall is gram positive, the thick wall of peptidoglycan layer. The gram negative bacteria, thin wall of peptidoglycan layers. This is only difference of gram positive and gram negative. The few example of bacterial everyday in life. One is Staphylococcus aureus, gram positive, spear, live on skin, can cause staphylococcal infections. Another one is E. coli or Escherichia coli, gram-negative rod live in large intestine, can cause food poisoning. Another one is Lactobacillus acidophilus, gram-positive rod live in gastrointestinal tract, found in fermented foods. Another one is Streptococcus mutans, gram-positive live in mouth and the contributed digestive areas. Another is Pseudomonas aeruginosa, is a gram-negative rod live in soil and water, can cause antibiotic resistant infections. This is a few examples of gram-positive and gram-negative bacteria. The first step, preparation of gram staining. What are the procedures of preparations? The prepare for laboratory works. Example, put on glove and tie back long hairs to prevent contaminating the bacteria sample. You will be testing the disinfectant in your workspace under the fume hood or in another well ventilator areas. Check that the Bunsen burners, microscope are functional before the start the works. Second step, sterile a glass microscopic slide. It is a glass slide is tried wash in sterile distilled water to remove the grease and dirties, and disinfect the slide with ethanol, glass cleaners and white cloth, whichever method is recommended by your laboratory. Third steps, add a sample to slide. You can use gram staining method to help identify the bacteria. Present the medical sample or bacterial culture grow in a petri dish. In order to gram staining to useful, a thin layer of sample on the stain. The sample under 24 hours old is recommended. And as old bacteria maybe have damaged cell walls that responds less predictably to gram staining. If using the tissue samples, add 1 to 2 drops on the glass slide. The spread evenly on the slide to the form of thin smear. Use the edge of second sterile glass slides. Allow it to air dry before continuing. And after sterile inoculum, the loop, Bunsen burner is flame until the grow. And then it let cools. The use to the place drop of sterile waters on the slides. Then sterile cool the loop again before the transfer of a tiny sample of bacteria and gently stirring into the water. And another bacteria is broth. Example, without agas, the broth should be mixed. Texture that added an inoculum and above without the adding of extra waters. If you have the swab samples, roll the swab lights lightly across the slide. And heat fix of smears. Heat will fix the bacteria to the slide, so they are not and easily rinsed off during the stains. Quickly pass through the slide 2 to 3 times through the Bunsen burner flame. On the heat of the top of electric slide warmers, do not overheat. The microorganism will be dead, or the sample may become destroyed. If using a Bunsen burner, the flame should be small blue cone, not a tall orange one. So the top of the flame is suitable for heating. The alternative smear may be fixed by the methanol instead by adding of 1 to 2 drops of methanol onto the dried smears, draining off excess methanol and allow to it air dry. This method minimizes damage to the host cell, giving the clear backgrounds. This is the flame of the Bunsen burner is a blue in color, is suitable for heat fixing. And after the position of the slide on the staining tray, there is a number of only 20 slides the holders. The stain tray is show metal, glass or plastic dish with a small mesh or wire supported running across the top. Place to the slide on the top of the support to be liquid to be will using can prime down into the tray. If you do not have a staining tray, the slide can be placed directly to the top of the plastic ice cube trays. Second part of the gram staining, the perform the gram stains. The flood the smear with the crystal violet is the first counter stain. Use a pipette to flood the bacteria sample with several drops of crystal violets dye, sometimes called Gentians violets. The wait 30 or 60 seconds. The crystal violet dissociated the aqueous solutions into Cv plus and chloride minus. The ions have been ion penetrated through the cell wall and cell membrane are both gram positive and gram negative cells. Cv positive ions, they interact with the negatively charged components, the bacteria's cells, to the stain of the cell purple. And rise of the crystal violet gently. Till the slide use a wash bottle to squeeze a small stream of distilled or tap water. Over the tap water slide, the water should be run down over the surface of the smear, but not aimed directly. Right? Do not rise excessively, which might remove the staining from the gram positive bacteria. And finally, flood the smear with the iodine, then rise. Rinse. Use a pipette to cover the smear with the iodine. Let it sit for at least 60 seconds. Then the rise of within the sample carefully. The use methods, the iodine form of negatively charged ions interact with positive, the form of large complex crystal violets and iodine negative complex, the inner outer and layers of the cell. This will be trapped the purple crystal violet colors in the cell. It has strain. The iodine is corrosive. Avoid inhalations or ingestions or skin contacts. This is very cautious. The iodine solution used very carefully. And add a decolorization. Then the rinse quickly. The 1:1 mix of acetone and the ethanol is typically used for critical steps, which must be timed carefully. Hold on the slide at the angle. Then add the decolorization until no more violet colors is visible on the trained runoff. Typically take under 10 seconds. Or even less time for decolorization containing highly concentrated of acetone. Stop immediately or decolorization will be remove the crystal violet stains from both gram positive and gram negative cells, and stain will have to be repeated or immediately rinse off excess decolorization using the earlier techniques. Pure 95% plus acetone can be used instead. The more of acetone is the faster decolorization will works. They require more precious time. If we have the trouble, the timing is considered adding a decolorization drop by the drop. So one by one clearly added. And the flood is smeared with counter stain, then rinse. The counter stain typically is safranin or fuchsin, commonly used for safranin stain. It is used to blood red color to add extra contrast between the gram negative and gram positive bacteria. By staining, decolorization, gram negative bacteria pink or red in colors reflected. Leave it on for at least 45 seconds, then rinse off. Fuchsin will be used to the same maybe gram negative bacteria, more intensely used as such as Haemophilus species or Legionella species, may be the better option for beginners. Dry to slide, maybe allow the slide to air dry or plant to the dry using the bibulous papers. The sold for purpose, the gram staining is completed. And prepare the light microscope. First of all, the microscope will be properly cleaned in white cloth. Place the slide under the light microscope. Bacteria vary greatly in size. The total magnification required will vary from 400x to 1000x. But the basic microscopic 10x, 100x, 45x and oil emulsions. At the greater clarity, the place drop immersed the oil or slides. Avoid the motion during the application to prevent bubbles. So very carefully mount the cover slip. Move the microscopic direct so the objective lens the click into the place or touch of the oils. The oil emulsion can be only to use on specially designed lens, not a dried lens. This is the comparative positive and gram negative slides. Identify the gram-positive and gram-negative bacteria. Examine the slide under the light microscope. Gram-positive bacteria appear purple in colors due to the crystal violet trap within the thick cell wall. And gram-negative bacteria appear the pink or red in color since the violet was through the thin cell walls. Then the pink counter stain, they enter them. It is a sample too thick, maybe if false positively results. The stain a new sample if all bacteria type of gram positive to make sure the results is correct. If the decolors ran too long, we may be false negative results. Stain new sample if all bacteria type of gram negative to double check your results. So immediately to perform, right? And look up reference image. Example, if you are not certain what the bacteria is, look through the collection of reference image sorted by shape and result of the gram strains. You can find database on the National Microbial Pathogen Database, NMBT. And many other sites to be identified easier, commonly or diagnostically important example are sorted below the by gram status or shape of the bacteria. And identify gram-positive bacteria by the shape. The bacteria are further clarified by the shape under the microscope, most commonly as cocci or spherical in shape or rod in cylindrical shape. Here are few common gram-positive purple strains. Bacteria are organized by the shape. Gram-positive cocci, generally staphylococci. The mean of cocci is clusters or grape shape. Streptococci, mean of cocci in chains. Few examples of gram-positive rod then includes Bacillus, Clostridium, Corynebacterium, and Listeria. Actinomyces species, a rod of have been branched or filamentous sections. This is a gram-positive. First one is the purple in colors, gram-positive organism. Pink or red in color, gram-negative bacteria. And to evaluate mix the results. Some of the bacteria are difficult to strain precisely due to the fragile or waxness of the cell wall. They may have mixed or purple or pink strains, same cells or among the different cells of the same simius. Any bacterial sample over the 24 hours, the old culture will be a problem. The microorganisms already way to death. But some species are difficult to strain at any age. They may be require more special tests to narrow down for the identification, such as acid-fast stain, culture grow observation, or TSI medium culture, or genetic testing. Few examples of Actinomyces, Arthobacterus, Corynebacterium, Mycobacterium, and Propionibacterium species are all considered gram-negative positive bacteria, but often appear in conclusively strained. The similar and slender bacteria such as Treponema, Chlamydia, and Rickettsia species are difficult to gram stain properly. Dispose of the material. After using or after identifying the microorganisms, the bacteria are very vigorous and some of the pathogenic microorganisms. So the waste dispose procedure vary between the laboratories, and according to the material being used, typically by the liquid in the strain tray, is disposed of sealed bottle as hazardous waste. Soak slides in a 10% of bleach solutions, then dispose of them in sharp containers. Some of the questions. What is the most important or crucial steps in strains? What is the purpose of the gram straining procedures? What is the straining, and explain the procedures? In a cell, what is the thing that can be known by gram staining? What is the purpose of reagent of the gram staining? The few references will be provided. So please refer to our PPT presentations. Thank you so much for patient listening. Thank you. My next videos in short period of time. Thank you. Thank you guys.